So, welcome to Organic Matter. So, an organic compound is any substance that contains carbon, with the exception of cyanide, cyanates, and carbon dioxide, and its relatives are not organic but contain carbon. So, basically, an organic matter is matter that has come from a once living organism and is capable of decay or is a product of decay. So, it all has to do with decomposition. So if we think about carbon and our carbon sources, again, these are in gigatons of carbon per year in terms of fluxes. You can see that we have a lot of carbon, not only in the atmosphere, but much more is in the vegetation, soil, and detritus, as well as in the deep ocean, the surface sediments. Again, some in our actual marine biota, but most of it, again, is in the soil detritus pools. And again, with gross primary production, we have carbon going up, carbon going into vegetation, and respiration going back into the atmosphere. And so those are our big, and the same thing with from the ocean surface and back. But again, we have this huge fossil fuel source. So that's where we're getting the the CO2s into the atmosphere is from these fossil fuels, where you can see normally gross primary production and respiration are pretty close linking in terms of carbon fluxes. So we talked about, again, organic matter being the product of being something that can decay or the product of decay. So again, decay happens by microorganisms. So the most important function is the breakdown of organic material. That is a process in which the limited supply of CO2 is replenished. Again, with fossil fuels, it's not so limited, but normally it's this balance of primary production, respiration, and the microorganisms basically take that CO2 back into the environment. So there's five major groups of microorganisms in the soil. So it's all of these. We mainly talk about bacteria, but it's not just bacteria. There's protozoa, there's algae, there's other bacteria that are microorganisms that are part of this degradation process. And again, when we're talking about how much bacteria, we're talking about a large amount of bacteria in just a gram of soil. So if we look at the classifications of microbes, again, we have these soil microbes, and the idea is some of them get their energy source from chemicals, and some of them get their energy source from light. So heterotrophs, so these are chemoorganotrophic, require organic nutrients to serve as the source of energy and carbon. So fungi, protozoa, and most bacteria. So again, when we think about denitrifiers, for example, they're using nitrate as their organic tropes. If we think of autotrophs, they obtain their energy from sunlight or the oxidation of inorganic compounds and through the assimilation of CO2. We have photoautotrophs, which again attain their energy from sunlight, so algae, some bacteria, higher plants. And then we have chemoautotrophs, which is obtained by the oxidation of inorganic materials. So our denitrifiers are our chemoautotrophs because it's inorganic materials, while our chemoorganotropes actually require organic nutrients to serve as their source of oxygen. So this decomposition rate is again going to be influenced by oxygen, by moisture, temperature, the lignin content, how hard it is to break up, the size of the particle material, the compound structure. Again, if it's in the lakes or ocean, waves can actually do some of that degradation in terms of just breaking it up, not actually degrading it, but helping it to smaller particles. And then animal activity can do the same, helping get it to smaller particles, so then it can be used by the microorganisms. So if we think about aerobic versus anaerobic over time, so if you have degradation, these decomposition rates, again, very quickly we get about 30% decomposition, and then that aerobic is going to continue to decompose to about 20% of the remaining material over a year or two, while the anaerobic is actually going to not decompose completely, just because it's a slower process. So again, in respiration, microbes oxidize organic material. So it's an oxidation process by using a sequence of different molecules as their electron acceptors. So this oxidation reduction potential declines as the environment becomes more anoxic and alternative electron acceptors are used in anaerobic respiration. And again, this changes the physical and chemical properties of the environment around them because it changes the type of sulfur, nitrogens, and iron compounds and produces methane. So when we think about oxidation reduction potential, this is really important. So we have an organic substrate. This is our electron donor. And so at the first, this time sequence, first we're going to have oxygen reduction. So we're going to use that oxygen first, and oxygen is going to be decreased. Then the next, in terms of the most advantageous in terms of energy potential, is going to be our nitrate and our manganese. And so our nitrate is going to be reduced through denitrification, okay? And at the same time, our manganese is going to be reduced as well. And so we're actually going to see manganese 2 plus being produced. Next, we have iron. And so iron is also going to be reduced. And after iron reduction, we have sulfate reduction, where again, sulfate, SO4 2 negative, is reduced and hydrogen sulfide is produced. And then finally, we have methagenesis, where actually CO2 is reduced and we actually get methane CH4 that's produced. And during all of this, as our organic substrate is decreased, the amount of nutrients that are available in the environment increase. So we get an increase in dissolved nutrients as the degradation of our organic substrates go down, and we get an increase in methane and CO2 as the oxygen is used up and the environment becomes anaerobic or anoxic. And so again, we have our organic carbon, and again, we can see these relative voltage. These are the oxidants. And so the most favorable oxidant is oxygen. And so we're going to kind of go up this chart in terms of what we're going to use: the nitrate, manganese. Again, we have alternative sources of nitrate. We have iron, and then finally CO2, which is used in methane production. So if we think about a reduced soil level, so this is in a treatment wetland, for example, or just a natural wetland. Here you have your surface water here, and you have your roots. What you're going to find is if it is actually an anoxic environment, if it is without oxygen, then we're going to see our manganese is actually going to increase. Our iron, our reduced iron is going to increase. Our sulfide production and our redox potential is going to go down. It's going to go negative as the oxidation reduction potential goes from a positive to a negative, and we see different chemicals being respired. So when we think about organic degradation, the carbon-nitrogen ratio is extremely important. And so we already talked about the carbon-nitrogen ratio, but again, in that carbon-nitrogen ratio, if it's greater than 30 to 1, so 30 parts carbon to 1 part nitrogen, it's going to be nitrogen limited, and you're not going to see as much degradation take place because the microbes don't have the nutrients that they need. Again, 20 to 30 to 1, that's a good carbon to nitrogen ratio for the microbes for degradation. If we see less than 20 to 1, then that means that there's a lot of excess nitrogen, but we may not have the availability of carbon, maybe what's limiting the organics. So mineralization rate. Now this is, again, this carbon nitrogen doesn't say anything about the quality of that carbon. So if it contains a lot of lignin, then it's going to be much slower degradation rate. And as it starts to degrade, that carbon nitrogen ratio is going to come together, where again, we use that organics and we have more nitrogen being released in the environment. So if we think about soil microbes, again, they make up a relatively small portion of the total organic matter available in soil. But again, it functions as a catalyst for the transformation of C and other nutrients. Most of the soil organic is contained in the non-living components, so plants, animals, microbe debris. And so cellulose is the largest portion of this fresh organic material. And it decays rather quickly, but again, it needs that nitrogen, that CN, to decay. Lignin is going to degrade very slowly. But again, it takes a long time to degrade, so you need more alkaline solutions for that degradation to occur. So if we look at kind of our cellulose, that's going to be very rapid. Our hemicellulose, again, this is a ringed compound. So it's going to be moderate to slow, especially compared to cellulose, which is a rapid chain degradation process. And then our lignin is going to be slow. Our proteins are going to be fairly rapid, but it's a smaller composition. And then finally, our polysaccharides, again, our starches and pectins, are also going to be degraded fairly rapidly during the decomposition process. And so if we look at the original composition, again, our cellulose and our hemicellulose is going to degrade rather rapidly. And again, that's a large percentage of our organic matter, but our lignin is not going to degrade as quickly. So as this decomposition proceeds, the water soluble fractions, so these are sugars and starches and pectins, they're going to be rapidly utilized by the microflora. And then the alcohol soluble fractions, the waxes, the fats, the resins, the hemicellulose, they're going to decrease with time as they're being utilized, but the lignin is going to kind of persist and accumulate in the decaying biomass. And so decomposition rates are also proportional to the lignin content. And some researchers have said that maybe lignin may be more reliable of the decomposition rate that's expected than necessarily just relying on the C/N ratio. So C/N ratio is also extremely important for the composting process. So we're going to talk a little bit about composting. So what does compost need? This is now an aerobic process. So it needs water, it needs oxygen, it needs microorganisms, it needs feedstocks. And then what it gives off, it is actually evaporation. So it gives off water, it gives off CO2, it gives off heat, and hopefully it doesn't give off odors if you're composting correctly. And then you have a compost product that destroys pathogens and can be used as a nutrient and soil amendment. So again, the organisms, these are microbes, they need kind of what all living things need. They need food, they need oxygen, they need nutrients, they need water, and they need a hospitable environment in terms of temperature and neutral pH conditions. So again, they secrete enzymes to dissolve those organics. And so these enzymes they break down the chemical bonds. And so again, we have not just bacteria, but it's also bacteria, fungi, and the Actinomyces. So the types of organic, just as we talked about before, in the composting process is going to be the same thing. The sugars and starches are going to go really quickly. Then the fats and proteins, a little bit slower, are going to be the cellulose, hemicellulose, and chitin. And finally, the lignin and lignocellulose are going to be resistant to decay. So in this process of composting, again, this is over time. At the beginning, it's going to go thermophilic. So this is actually, it provides its own heat. So we're going to talk later about anaerobic digestion. We actually add heat because it's an anaerobic process. It's not as energy efficient. And so it doesn't heat itself up. The composting process itself is very hot. It actually provides its own heat because it's so energetically favorable. And so the compost is going to start, let's say it's 68 degrees Fahrenheit, 20 degrees Celsius. And it's going to come up and it's going to reach this thermophilic stage over time. And then it's going to be cured and matured until it goes back to ambient temperatures. And so as it reaches these thermophilic temperatures, we get most of our pathogens killed. If you get a little bit higher up to the 150, then we're going to get a lot of our seeds killed off, which we want because we don't want seeds that are weeds to go into our compost if we're putting on agricultural fields. But if we go too high, we're going to sterilize it and kill off our good bugs. So we don't want to do that. And so what usually happens with a compost process is you actually turn the compost every now and then, and that actually helps with the degradation rate. But it is going to mean that the temperature is going to go up, you're going to turn it, it's going to go back down, it's going to start the process again, turn it, go back down. So that cycling effect is actually good for the process. And so we do turn, and you do see the temperature kind of go up and down during that turning process. And again, you can see here they turned it about every 10, 15 days. So in the first part of the compost process, the bacteria again are going to break up the cellulose into glucose, sugar, starch, and proteins. And this makes the temperature pile go up. And again, you can produce endospores as the pile heats up, that bacteria can then reactivate as the compost process continues. In phase 2, that's when the thermophilic bacteria and the fungi take over. So again, the other ones have these endospores and they go dormant, but these pathogens are killed, the carbohydrates are broken down, but the hemicellulose start to decay and the temperature continues to rise. In phase 3, now our temperature's going down. The mesophilic microbes return. Again, we're going to see the metabolic activity decrease, which makes the temperature decrease. And then the lignin, again, they again start to decay some because again, we've given enough time. And we should get arthropods that start to forage. So if you actually haven't gone through the thermophilic stage, you don't want your snails and your arthropods and your slugs and centipedes to come in. But once it's cooled down and you've killed off your pathogens, those actually help with further decomposition. And so here again, we have our dynamics during. So here's our temperature. So again, we hit that thermophilic stage, which is great. And we have all of these, here's our temperature. So we have all this bacteria. Again, as the temperature rises, the bacterial rate goes up as they are happy and growing. And then again, we have our fungi, which actually kind of hits here, and they start to go up. And then again, you see these numbers. So these are, again, the numbers of bacteria. So we see a big dip during the thermophilic, and then we see, again, another dip during the mesophilic. As the temperature goes up and down, that bacterial count is going to mirror the temperature. So the key processes for composting, first of all, is the initial feedstock mixture. So you need to think about the organic matter. We obviously need organic matter for the microbes to break down, the nutrients, the CN ratio, the degradability, the moisture, the bulk density, how is it heterogeneous? And then also you want to think about contamination costs, regulations, availability. So again, our ideal range, as we said, for CN ratio is about 25 to 35 to 1. Again, if it's high CN, that means that we're N limited, and low CN means that we're carbon limited, and we might get extra nutrients. That nitrogen might be released as ammonia gas. Particle size and shape is very important. So the smaller particle size, if you think about this being a particle size, if it's broken up into smaller pieces, like from wood shavings, wood chips into shavings, then the microbes can get at that surface area better. So we want to have some both rigid structure, so we have airflow, but we also want small structure so that the microbes can get at those particles. So if you think about these core particles, we need pore spaces between those pore particles for airflow and for the microbes to have that surface area. And there ends up being kind of a liquid film around it. And then our free airspace helps with that low flow. So what we don't want is we want a low bulk densities where it's not compacted. We don't want a high bulk density where it's more densely packed in there, because then we've lost some of our pore volume. So the key process is, again, we need to think about our pile moisture. So we want it to have a good moisture content, but we don't want it to have too much moisture. So our optimum is about 45 to 60% moisture. If it's too much moisture, then we're actually not going to get, we're actually not going to have enough, we're going to have anaerobic conditions start to form. And, but if it's too dry, then we might actually get dust problems. So we want it to be about 50% moisture content. For aeration, we need it to have oxygen. This is an oxygenated process. This is not an anaerobic process. We don't want methane and hydrogen sulfide produced. So ambient air is about 21% oxygen. And so we're going to provide air. We don't need to provide full oxygen. We're just going to provide air. So we can actually pump it into the piles. As you see here, there's air pumps, or we can just turn it so that we have that oxygen in the environment. And so again, the compost organisms can survive with as low as about 5% oxygen. But again, below 10%, they might switch to starting anoxic and anaerobic conditions, and we might get hydrogen sulfide, which smells, and we don't want that. So our aeration is going to be controlled by the porosity. Again, we want some big material to kind of get us bulky and get those air spaces in between. We don't want it to be too compact, and we don't want it to be too moist. So we can either do it with blowers, or here you have blowers inside the compost pile, or just rely on convection. For the pile shape and size, smaller piles are going to have greater airflow, especially in the middle of the pile. We don't want the middle of the pile going anaerobic, but if they're too small, then we're not going to get that high temperature that we want for the pathogen kill. So again, and if it's too big, it's going to be too heavy and compact. So you can only really have a really bigger pile if you have a higher carbon material, a low bulk density and you have like structure to kind of keep it up. Otherwise, you want to do a smaller pile but still big enough to maintain those internal temperature. And again, depending on the equipment they're using for turning, that needs to match your pile size. So we also want to think about the shape of the pile. So again, this would be a good shape because again, the rain as it comes down, it's going to go off the pile. What we don't want is the rain to come into the pile and then penetrate and get anaerobic conditions inside of our pile. In terms of temperature, again, higher temperatures, faster breakdown. So if we are greater than 160, though, we actually are, it's too high. But again, most weeds and pathogens are killed at about 130. So when we think about the process to reduce pathogens, again, we have a certain time and temperature requirement to reduce those pathogens. For an aerated static pile or an in-vessel composting unit, we need to be at 131 degrees Fahrenheit for at least 3 days. If it's a turned wind-run system, it's not static, then we need to be at 131 for 15 days or more within 5 turnings. And so again, this is this 131. So to kill pathogens, we need to have that 131. And depending on how we manage the pile, it has different days that it needs to have that temperature to kill off our pathogens. So again, it's going to be mesophilic for a few days to 2 weeks. Then it's going to be thermophilic for 3 weeks to several months. And then it's going to go back to curing and maturing for 1 month to several months. So again, we're talking about a months long process. We're not composting in 5 days. So when is the pile done? When the temperature is about less than 10 degrees warmer than the ambient temperature. That temperature has now gone back down. It's about a third of the original size. You can't recognize the original materials. We don't see food. We don't see broccoli pieces. It smells earthy, not like ammonia or rotten eggs, but it has like a nice earthy smell. Again, it has stability, and then you do lab tests for doneness. And so again, when we think about the original versus the ending range, moisture content, we wanted, again, anywhere from 40 to 60, preferred is 50 to 60% moisture. Again, this is our preferred organic content. We don't want to be carbon limited, and we know that it's going to go down, but we want to make sure that it doesn't go below 20 to 1. We want at least 5%, but more likely 10% oxygen. Our temperature, again, we're going to go between 120 and 150. 160, we don't want to go above 160, but we definitely want to get above 131. Our pH, again, around 7, around neutral. Our particle size, again, it really depends, but we want it to be about a 1/8 to about 2 inches. We don't want it to be all really fine, and we don't want it to be all really big. We want a mixture. And our bulk density, again, if we think about 0.7 kilograms per liter, that's what we want our bulk density. We want it to be a little fluffy so that we have about 50% free airspace in our pile. So we can manage them differently. We can manage it. Here is windrows. Here's a windrow turner. We can have it in static piles here. We can have in-vessel systems. Here's some other in-vessel systems. We can have covers. We can have it where it's we blow air into it. So there's a lot of different types of systems for composting. Often after composting, we will have screening where we'll get like plastic bags out, pieces of metal, anything that, or any pieces that didn't get composted properly will be screened out. And then whatever's screened out usually is landfill, but it's again a small portion. It's usually plastics and things. So again, if we look at kind of the critical control parameters, we want to delivery inspect the input, and we need to make sure that, especially when we're bringing the food waste to our site, if we're composting food waste, that we are dealing with it and we don't have odors and smell from the storage of that food waste. And we don't want birds or vermin to get into before we start the composting process. Then we'll have 1 to 4 weeks of thermophilic disinfection so that we meet the pathogen time temp limits, eliminate our volatile organic carbon so we don't have smells, eliminate our weed seeds, our vector attraction, and then we're going to have 4 to 8 weeks of curing and maturing. And this is where we're going to have nutrient stability. And then we're going to dry and screen, and then finally sample it to make sure that we meet our pathogen reduction limits, and then we can prep it for whether it's in bags or in bulk for selling. So all of our compost guidelines are, again, the US EPA has 503 guidance on testing methods and for compost and composting. Um, and there are things too in terms of we want to do organics. The USDA has organics programs for composting and what we can put in. So again, we can't put raw manure within 120 days of harvest where edible portions contact the soil directly, but not within 90 days. We're just going to put raw manure. But again, if we have compost, then we need to look at the initial ratio of what we start with and the temperatures that we get to. That's what they test for in terms of using it as an organic product. If we think about again for the FDA, the Food and Drug Administration, so there's all these different rules. EPA has the composting thing, the USDA does organics, the FDA has rules too. So raw manure is not without a year of something that's edible for produce that's eaten raw. Compost is going to do that EPA rule in terms of as long as you meet those things, then you can apply it. Again, testing, you're supposed to use process control to achieve those pathogen destruction. And so we're looking for salmonella, E. coli, we're looking for all types of things, and there's different types. So again, less than 3 most probable number for E. coli, less than 0.3 most probable number per gram. So again, depending on the species, we have limits that we have to meet for that Section 303 composting permit. So again, our benefits of composting is the retention of nutrients with this organic soil matter that we can put onto the soil so that we have nice fluffy soil for growing crops. Reduced N2O because again, instead of adding fertilizer directly, because we balanced it out with the organic material, the idea is that we get less N2O and denitrification that takes place, enhanced soil fertility, decreased bulk density, reduced soil erosion, and the eutrophication that takes place from soil erosion, and improved water holding capacity for crop systems. All right. Thank you very much.